Good evening, everyone. Namaste. I am Kruti Thakore, and I am a certified Ayurvedic wellness practitioner. I would like to welcome you all to this GCAR General Club June 2025 meeting. GCAR stands for Global Council of Ayurveda Research. General Club is an educational initiative by GCAR where we discuss published research articles and critical analysis to stay updated with the latest research and promote evidence-based Ayurveda. GCAR is an international non-profit organization based out of USA with a sister chapter in India by the name of Ayurveda Anusandhan Abhiyan Foundation. The mission is to encourage, educate, facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence-based health science globally. And you can learn more about us at www.ayurvedaresearchusa.org. Please consider joining us as a member, which will bring you access to all our resources. As you all know that no non-profit organization can succeed without its support of volunteers. So we would highly appreciate if you would consider joining us as a volunteer. Also, no non-profit can operate without the support of the speakers which we have today and the volunteers. So please join us, and we can place you in variety of projects based on your skills and interest. Now I would like to introduce you all to the eminent speaker. Akshat Thakore is a senior research scientist at a leading global consumer goods company where he focuses on advancing research in skin health. He holds a bachelor's degree in biomedical engineering from Rutgers University and a master's in applied biomedical engineering from Johns Hopkins University. With strong foundation in translational research and passion for improving everyday health outcomes, Akshat brings a unique blend of academic expertise and industry insight to his work. In this talk, he will explore the role of Ayurvedic herbs in modern skincare science and their potential to bridge traditional wisdom with contemporary dermatological research. Over to you, Akshat.
Thank you so much for that introduction. So, hello everyone, welcome to today's presentation. Like the introduction said, I'll be talking about the role of traditional herbs and how they can now be applicable in modern skincare as well as modern formulations using modern science. So how do we bridge that gap between what traditionally we have been told about these ingredients and their benefits, and how do we bridge that gap to the consumer who may not be familiar with these traditional benefits, but explain it to them in a modern way with clinically backed science that can then speak to the actual benefits of these ingredients. So what I'm going to do is I'm going to share my screen so we can start going through the presentation. And if I can just get a thumbs up that everyone can see the screen, if you can just react.
We can see the screen.
Perfect. So the topic for today is going to be Ashwagandha and skin health. So Ayurvedic heritage meets modern science. So the particular herb that we're going to be talking about today is Ashwagandha, like the screen says. And it's an exploration of how this ancient herb delivers clinically validated benefits for modern skincare formulations. So a quick background, Ashwagandha, also known as their scientific name, Wittenia somnifera, is classified as a rasayana, or a regenerative therapy in Ayurveda. Traditionally, it was used to promote skin vitality, longevity, and youthfulness. And now, based on the modern science, we have multiple peer-reviewed studies that also validate a lot of ashwagandha's traditional uses for modern-day skincare. So things like increased hydration, elasticity, reducing wrinkles, fine lines, and also helping with inflammatory markers. So think redness or itch is a benefit that can also be delivered through this ingredient. So I'm going to cover 3 different papers today that all talk about ashwagandha and its effectiveness in clinical settings or in formulations. The first paper was a randomized double-blind study. It was actually from 1999, but it's still applicable today based on the techniques they used to test the ingredient as well as the way they went about testing it. So this was a clinical study presented where they had two sets of groups. One had the placebo, the other one had the ashwagandha-based formulation. And what they saw was when they measured it using a corneometer, which is a traditional skincare clinical tool to measure skin hydration levels, they saw a 15% increase in skin hydration for the ashwagandha formulation group compared to the placebo. There was also a statistically significant decrease in wrinkle reduction, specifically wrinkle depth. And the most important part was that there was zero adverse effects, proving that it was safe for daily topical application. The next paper that I'm going to be talking about is speaking on the anti-inflammatory benefits of ashwagandha. So ashwagandha is a very potent anti-inflammatory ingredient. It has been proven in several research papers, specifically in what we call in vitro research, where in a lab setting, you might be using skin cells or different fibroblasts or keratinocytes, which are different types of skin cells that you typically use for skin health research. And what they do is they treat the cells with a pro-inflammatory control to give a inflammation or an inflammatory response. And then they treat it with the potential treatment ingredient to see if there is a reduction in those inflammatory cytokines or those particular markers that you're looking for. So some of the markers that you might look at are things like IL-6 or TNF-alpha. These are just two of the many inflammatory cytokines that are present. So what they saw was when you treated the cells with a ashwagandha extract, you see a significant reduction in these pro-inflammatory cytokines, as well as a suppression in another cytokine, NF-κB, which is critical pathway in skin inflammation. So you see a suppression of that as well. This then shows you the potent anti-inflammatory aspect of ashwagandha. So how do you talk about inflammation, and then how does it translate to a skin health benefit? So most of the time, if you're using a skincare product, specifically if it's a lotion or a moisturizer, things like that, you are not going to just see, this helps with inflammation on the pack, right? So what kind of language do you look for to differentiate or figure out if the ingredients in there are going to be suiting to your needs? So if you have redness, if you have itch, you have bumps, typically you have inflammation in those areas. So if you have bumps on your face, it is a sign of increased inflammation. If you have itch, it is a sign of increased inflammation. There are also several disease or compromised skin barrier states, such as psoriasis, atopic dermatitis, dry skin, eczema. These are all inflammatory skin states. So you need an ingredient that helps negate some of that inflammation that is going on, or some of those flare-ups. The language you look for is when companies say things like soothing, or it helps with itch, or it helps with redness. Things like that is what you're looking for. Companies are not going to put on pack, this helps with inflammation and things like that, because that's typically a drug claim, and that's something you would see from a medicated product. But if you don't really have a disease or a prescription from your dermatologist, you are not going to get a prescription for a medicated product. But if you have some minor itch, you have some minor redness that you want to address, you can still find a product that addresses those needs by looking for those keywords like soothing, redness, itch, and things like that. The next paper was again published very recently, 2023, where they looked at the impact of ashwagandha and enhancing the skin barrier. So the first thing they looked at, which was TOOL, which stands for transepidermal water loss. This is the rate of water escaping from your skin barrier. So think about all the water that's below the surface of your skin. How quickly is it leaving the below surface, reaching the top layer, and then evaporating out into the environment? How quickly is your skin drying up? The next thing that they looked at was hydration levels. So not only do you want to stop transepidermal water loss, but you also want to increase hydration. That's how you know your skin is properly hydrated. It is supple, it is moist, it is doing good. And then the final thing was looking at elasticity, to look at skin firmness and bounce. How plump is it? Because the more hydration or the more water content you have, the better your skin will have its bounce and firmness and its radiance. So what they saw was with the ashwagandha treatment, you see a 20% decrease in TUL, also known as transepidermal water loss. You see an 18% improvement in skin moisture content, as well as a significant improvement in skin firmness and bounce. So it's a two-pronged approach, right? We're stopping the water from leaving our skin barrier, while we're also increasing the water content or the hydration in the skin surface layers. So a quick recap of the three papers, as well as the traditional claims that you would have surrounding this ingredient. The first would be that rejuvenation claim, right? How does this ingredient help with rejuvenation? So we see the clinical evidence. We see increase in elasticity. We see a decrease in wrinkle depth. The next is anti-inflammatory. We see that with the reduction of those pro-inflammatory cytokines. And then the final one is improving the skin barrier or repairing or strengthening the skin barrier, which you see from that reduction in TUL and that increase in hydration. If you reduce the water loss, you increase the hydration, your skin naturally becomes less dry, flaky. It improves the moisture content in your skin. Your skin, it has that bounce, and that also helps with the skin elasticity and the wrinkles that are there. So now that we see the clinical evidence for ashwagandha, we know that it works. We see multiple research papers that are talking about or highlighting the multiple benefits that it has to your skin. How do you choose the product? How do you find products that have it? The first thing you want to do is on the ingredient list for whichever products that you are looking at, whether it's a toner, a serum, a lotion, you want to look for the name. Now, they're not going to say ashwagandha. Most ingredient lists are always in the scientific name. So you want to look for the exact scientific name, which is the Wittania somnifera root extract or Wittania somnifera extract or any variation of that Wittania somnifera. That's the key thing you want to look for. Once you see that on the ingredient list, the next thing you want to check is where in the ingredient list is this ingredient? Is it near the beginning? Is it near the end? Things like that. Typically, if it's one of the last ingredients mentioned on an ingredient list, it typically means that there's not that much of it in the product. The way companies list their ingredients, it's always listed in order of weight by volume. So the ingredient that's in the product, which is at the highest concentration, will always be found at the top of the ingredient list. So a lot of times you'll see water or glycerin or things like that near the top of the ingredient list because most of the formula is made up of that one particular ingredient. So typically, if you're looking for a product that has ashwagandha in it and you want the benefits of ashwagandha, you want to make sure that it's somewhere near the middle to the top of the ingredient list, because that means that they have put a significant amount of the ingredient in the formula, which will then deliver the benefit that you're looking for. And the other thing you can also check for is synergistic herbs. Because this is an anti-inflammatory ingredient, because it helps with radiance and things like that, it really pairs well with some of the other ingredients that deliver similar benefits. But things like aloe, turmeric, ginger, as well as some of the anti-aging ingredients that you might often see on lists, which is like niacinamide or hyaluronic acid. All of these ingredients work really well together because they all deliver similar benefits through a different mechanism of action or a different pathway in the body. So, hyaluronic acid is a very popular ingredient these days. We know it helps with skin hydration levels. We know how it works is that it helps with retaining water content in your skin once you have hyaluronic acid there. The ashwagandha will also help with hydration levels. So the two ingredients can go really well hand in hand. So things like that is what helps you identify the different products that you're looking for. There are several brands that are already using ashwagandha out there. So for example, Selena Gomez has her own brand called Rare Beauty. Several of her products have ashwagandha in them. I think one of the face creams, one of the hand creams have it in there. On their website, sometimes they even call it out by saying it helps with inflammation and redness and things like that. So it's very easy to just do a quick Google search of skincare products with ashwagandha, and a lot of them really start talking about the benefits of these products that are there. But of course, you also want to find products that are clean. You want to make sure that they are natural, they're organic, they don't have too many chemicals, things like that, things that can irritate your skin, because that defeats the purpose of using an anti-inflammatory ingredient. If you're looking for an ingredient that helps with redness, but then you're using a product that has other ingredients that will increase redness, it really doesn't really help you with the way you want it to. So there is a website called Inky Decoder, INCI decoder.com. If you just search on Google Inky Decoder, it will come up. It is a great tool for anyone who wants to know what is in their products. You go into that website, you type in the name of the product that you're currently using or thinking about buying. It will give you the name of the product. It will give you the full ingredient breakdown list. And what they have done is for each ingredient, they will tell you, is it a good ingredient? Is it a bad ingredient? How does it fit in terms of skin? So, they have certain ingredients which are called superstar ingredients. So think glycerin and things like that, some that are really good for your skin. And then there's others which are ranked as not so good, which might be like fragrance and things like that, which can sometimes irritate the skin. So it's a great tool to figure out the products that you're looking for and whether or not the ingredients are clean or good for your skin. And the final summary we want to talk about is the benefits that we're looking at for ashwagandha. So it's a four-pronged approach. The first, of course, is hydration. We see a significant increase in skin moisture retention, as well as preventing some of that water content loss. The next is anti-inflammatory. It reduces key inflammatory markers and pathways in the skin, helping to kind of soothe the skin, reduce redness, itch, things like that. The others is elasticity. We see an improvement in skin firmness and a reduction in visible aging. So your skin looks a little bit more radiant, looks a little bit more supple. And then finally, of course, the main important thing is safety. We see that in all of these studies, they have tested it for safety, for adverse effects, and we see that it was perfectly safe and it was a very good multifunctional botanical for skincare. So it kind of bridges that traditional benefits that we have seen in traditional literature, as well as now modern science that is backing some of that traditional claims to prove that it's effective, but also there's evidence behind some of the benefits that it talks about. And that brings us to the end of the presentation. I'll open it up for any questions if anybody has.
Thank you, Akshat. And we will open it up for questions. If anyone has questions, please go ahead. Yeah, Kavita, go ahead.
Akshat, this is Kavita and great talk. I think I missed the first 5 minutes of your talk, so I'm not really sure, but I just had a quick question. So do you know how this product affects different age groups? Has there been any age-specific data being collected?
That's an excellent question. I think most of the studies have been tested in the adult population in terms of the benefits and things like that. Because most of the benefits around this ingredient are tied into the anti-aging aspect of it, hence why they try to tend towards testing it into adults. But we have also seen other research papers, which I didn't cover today, where they have also talked about ashwagandha for use in topical formulations for young adults, as well as for babies and children. So again, it's been proven that it's safe in terms of topical application. The only call out that certain papers talk about is if you are taking an oral supplement of ashwagandha, there is a call out for if you are pregnant to basically consult with your doctor before taking ashwagandha, because there could be some potential side effects. But again, that is for the oral supplement. For topical formulations, the amount that these products contain is so little that the chances of it going all the way through your skin, into your bloodstream, into all that is slim to none. So typically topical formations are perfectly safe. It's just the oral consumption is one that would be called out specifically for women who are pregnant or trying to become pregnant.
Can I ask a follow-up question to this? So if it is oral supplement, was this tested for the skincare, the oral supplements?
That's a little bit of a gray area. Typically, the oral supplements is tested more for the stress relieving benefits of ashwagandha. So they're looking at improvement of sleep, improvement of mood, and things like that. For the skincare benefits, it's more beneficial to you if you find a product that is a topical formulation of ashwagandha rather than an oral supplement. The oral supplement will promote more for sleep and things like that.
Okay. Sorry, I have another question.
Yes.
So, talking about the anti-inflammatory markers, I'm an immunologist myself.
Yeah.
So how did you test these markers?
So typically in skincare,
given it's a topical application, how did you test this
typically the way the anti-inflammatory benefit of ingredients is tested in a skincare setting is through in vitro assays. So in vitro would mean in a lab setting, not in actual human skin. But what they would do is they would take skin cells, so whether it's keratinocytes or fibroblasts, so they would do cell cultures using these skin cells, and they would first increase the inflammatory response by adding in a pro-inflammatory ingredient. So typically a control, I don't remember the name off the top of my head, but there is a control that they use to induce a inflammation response. And then whichever ingredient you're trying to show anti-inflammatory benefits for, you would introduce that ingredient into the cell culture. And what you're trying to measure is that reduction in those pro-inflammatory markers. So you see a reduction in those cytokines like IL-6, IL-8, things like that. That is showing that you are having a reduction in that pro-inflammatory response when you introduce this anti-inflammatory ingredient.
So was this assays, do you have any idea what kind of assays they did? Was it like a Luminex or like a flow assay?
Yeah, so these are typically done in vitro human cell models, so with keratinocytes and fibroblasts. And this is where you would have basically a benchtop cell culture assay. Where you're basically just growing the cells and then introducing that pro-inflammatory marker.
Yeah, I got that, but you know what the output assays are? What kind of an assay they use to study these markers? Do you have any idea?
Yeah, I can look it up. I don't remember the exact name of the assay off the top of my head.
I work with a couple of derms myself, so I just was curious about it.
Yeah.
And I was just like looking into a panel with PBMCs and serums. So I was just wondering if there's someone who studied in serum to look into the effects of, ashwagandha in the serum samples of these patients, which would be a good output systemically.
Mm-hmm. No, that's a great point. I can definitely look at the paper real quick just to find the exact name of the assay.
Just curious.
Yeah, no worries.
Great talk though. Thank you.
Yeah, no worries. I'm just trying to find the exact paper because I saw it in the exact.
While Akshat, you're finding the paper, once you find it, you can put it in the chat. Dr. Shah also has a question for him.
Yes.
First of all, thank you, Akshat, for a very nice, succinct presentation. And for anybody listening in later on, I want to also thank Kriti because she's coming in as our new co-chair for journal clubs. So I'm Pratibha Shah. I'm the founder president of Global Council for Ayurveda Research. And we are in our, I think, 4th year of journal club meetings every month. So thank you so much. I just, again, for the benefit of people who are here today, and thanks, Kavita, for wonderful questions. For people who are here today and who will be listening in on our YouTube channel later, usually the format of the journal club is we pick up published papers and we present a summary and then we present a critique. So I was also curious about, did you have any critique for these papers? First of all, I would like to have links to these papers, which we can put on our YouTube annotation, Kriti. When the recording is uploaded, we can put the links because, again, for future, we like to share the links to the papers that are being discussed so that people can read and come for the interactive session for any critique they may have. Sample size was small or inclusion criteria was not clear, or there was a conflict of interest or anything like that, because that's what we are trying to encourage in our Ayurvedic fraternity is the critical eye. Not everything that is out there published is authentic.
Yeah, no, that's a great point.
Thank you,
Kavita.
Kavita.
In terms of critique or anything like that, I wouldn't necessarily say I have any major critiques. The main issue would be that because this is a traditional herb that's not necessarily known to Western scientists or Western researchers, the amount of research papers that you see from credible sources is relatively low. I think this is an area where we can still continue to expand our research into these traditional Ayurvedic herbs and generate more data for them. I think the data that we have currently is extremely promising. It's just the number of published papers or the number of published references that we might see is not as robust as what we would typically see for Western ingredients and formulations. So, if you look at the number of papers that are out there talking about the benefits of hyaluronic acid, they might be in the thousands. But when you look at ashwagandha, it might be only like 25, 30. This is still a relatively new space for Western researchers to go into. And I can speak from firsthand experience where even somebody who's working for a consumer goods company in skin health, while we are exploring new ingredients all the time, traditional ingredients like ashwagandha or turmeric or ginger, things like that, are not usually at the top of the list of ingredients that are being considered. While that pattern is slowly starting to change and we're seeing more and more of these ancient ingredients now coming over from the east to the west, I think we as researchers or we as a group can kind of promote the interest in these ingredients, so that way it gets on the radar of some of these researchers who are exploring these ingredients. People who are working in a R&D space now know the name of ashwagandha because it's now a trendy ingredient that's already being taken care of. But I'm thinking even of other Ayurvedic ingredients that we might have heard of, like manjistha and things like that, which is not really familiar in the Western world. I think we can do a good job at promoting these ingredients so that more and more people become familiar with them. And as the familiarity increases, the research itself will follow because more people will want to start testing it based on what they're hearing.
I think one of the challenges is the difference in the framework of the Western versus Ayurvedic. Ayurvedic is more whole thing, and the Western approach is let's isolate the active principle. And I think there comes a little fundamental, very important difference. And then also, let's say you were to do any research on ashwagandha. First of all, you need to find a standardized source for that ashwagandha. That is another problem because all over India, different areas ashwagandha will have different composition and qualities, significant differences. And another important thing is what part of ashwagandha is being used. There's a huge controversy in Europe right now because ashwagandha was banned in Denmark. I don't know if you're aware. And some other European countries also copy pasted that ban in their countries. And that concern is coming from primarily, I think, because of ashwagandha has become like a billion-dollar industry, multi-billion-dollar industry. People are now using leaves and stems and calling it ashwagandha, whereas traditionally only ashwagandha root is used. And it is used only in certain forms, not extracts, for example. That's not a classical way of giving ashwagandha. So what happens is now you are using ashwagandha non-classically, but your reference point is what is mentioned as a outcome in the text, and you're comparing like wrong things because if you use ashwagandha root and in the ways it is mentioned, like in powder, tablet, or ghee, or arishta, or some other classical form. And then if you don't get results, then you can say, okay, there is some discrepancy in what is claimed in the text and what is being seen. So I think these are the challenges of the science itself, because the model that we have right now, the standardized RCT, double blind and control and all that, it can be wonderful for testing a chemical. But when you're testing a whole herb, which has so many compounds, it has so many bioflavonoids or active ingredients, not just one. And then when it comes to polyherbs, that these challenges are further compounded. So that, these are some of the things to keep in mind when we are talking about research in ashwagandha.
Yeah, that's a good point. I think having that research of looking at the different parts and how each one compares. Is the benefit translated from root to stem to fruit or leaf? Things like that is definitely important, especially since it has become such a trendy ingredient and everyone wants to use it. In terms of, using a highly refined compound extracted from a root versus using, let's say, the root powder itself. I don't think there's necessarily limitations in that testing method, at least from what I have personally experienced or seen. The way we do internal testing in our labs, or I've seen in research papers, is the methodology is still available for you to test, let's say, a powder of a root versus a chemical compound. It's still relatively the similar methodology. I think the one thing that does arise from using let's say, an ashwagandha root powder is typically when you see favorable results, the first step that Western researchers want to do is, instead of using it in the form that tested well, they then want to start picking it apart and really diving in deep into, okay, this powder worked. Now let's find out what in the powder is the thing that's working so we can make a highly concentrated version of it and sell that to somebody. Instead of just going from, okay, the powder works, let's just use the powder as is.
Like it happened with curcumin.
Yeah.
Curcumin versus turmeric. What would have been the source of ashwagandha in these studies? Because that I know from direct experience that can be a big challenge. Like, where do you get it? What can be called as standardized source of ashwagandha for these kinds of studies?
Yeah, I think in all, if not 2 out of 3 of the studies, I think all of them were using a ashwagandha root powder. So it
From
was
where? Source from where
so
though?
so typically it
Company.
depends. If you're a researcher, you can reach out to cosmetic companies that are already having a commercial ingredient. So, for me as a researcher, what we typically do when we're doing developing of a product is we have a lot of cosmetic suppliers who supply us raw materials. So they have done all of the RMC, the safety testing, the tox analysis, all that of these ingredients, which they are then making available commercially for consumer good companies to buy to put into their formulas. So just to name a few, there would be a company like by the name of, for example, Givaudan. They're an international supplier of cosmetic ingredients as well as fragrances. You go to them, they have an ashwagandha powder. As a researcher, you can say, hey, I want to test this in my assay. Can I get a sample? They would send you a sample of that, because for them, it also works out because typically you would credit them in the paper where you got your sample from. They get some good data out of their ingredient that they can then sell to their customers. Like, hey, look, this research paper used our ingredient. It worked out really well for them. So typically there are commercially available ingredients that most of these researchers, whether they're college or companies, source from. And then they typically credit that in their paper that this is where our source of ashwagandha came from. And when they source it from there, the company is the one that has the reputation on the line to make sure that they're providing a good source that has been already analyzed for quality and control.
That makes sense.
So, Akshat, can you put the links to the research paper in the chat as well?
Yeah, I can put all 3 links. I already put one. I'll put the others as well.
Kavita, you have another question?
Yeah, sorry, I think.
No, go ahead. This is good.
Yeah. Akshat, I have another question for you. Pratibha, by the way, great question. I had that follow-up. So I was dividing myself into both the allopathy aspect as a scientist and also Ayurvedic. So I was going one after the other. But then before I hit about the root, I just see in one of the papers that they actually they started talking about the root and the leaves, but looks like they worked only on the root powder, the one which you had sent it out, Akshat.
Mm-hmm.
So my question is, they have tested this in the mice, right? It's very difficult to correlate with the humans when it comes to human studies. Although these are PBMCs, you're injecting the PBMCs into the mice. Of late, what we really see is when you look into the mice and compare to the human PBMCs, you're studying PBMCs just by itself is very different from injecting into the mice. Studying human PBMC cultures by itself is way different than when you inject into the mice. Because there's a lot of changes that happens. let it be at the genetic level or the nuclear level. It's very difficult. Even the phosphorylation changes a lot when you study human PBMCs by itself than when you put it into the mice. What do you
Yep.
say to that? Because these are mice studies. You can't really correlate directly with the humans. I mean, great talk. I'm not putting you off that. Great presentation. But then, it still bothers me when you show me mice data and talk about human PBMCs and their anti-inflammatory markers.
No, that's a good point. Typically, when you do animal testing, it's not necessarily only to show the effectiveness of an ingredient, but it's also typically to do for toxicity's sake, to show that it is non-toxic and things like that before
Sure.
you move to a human model.
Exactly.
Because typically you want to do it as a first step approach of, if it's not toxic here, then we feel comfortable moving into
QC.
humans.
Yeah.
Yeah, QC. So, but the paper the link that I sent, not only did they do the mice model in terms of not only toxicity, but also looking at the effectiveness of the ingredient, but also they did do human HA-CAT cells, like keratinocyte cells, to look at the cytokine or the mRNA expression levels of the pro-inflammatory cytokines. So that's typically also how it works in most skincare companies of research. Before you move into, let's say, a clinical trial, you do a in vitro skin culture assay to kind of do the QC of, if it works in the in vitro skin culture assay at so-and-so level, we feel comfortable increasing the level as we move into a clinical study. So just to give a rough example of how it would work is, if you're doing an in vitro assay, you might, let's say, for example, test an ingredient at 1%. As you move into a clinical study, you say, okay, it worked at 1% in an in vitro assay. Now let's put it at 10% in a formula for a clinical study, because typically whatever you tested in vitro, you do a 10x amount, roughly 10 to 20 times the amount in a formula for humans, or in a topical formulation. So typically in vitro assays are tested at a very extremely low level, might even be like 0.001%. And then you do a magnitude increase as you move into a clinical study. So here, not only did they do the mouse model, but they also did it in when they did a cytokine mRNA expression level in the
I see
the
that it's a
keratinocytes.
human cell line, right?
Yeah, a human cell
We did
line.
that in a human cell line.
Yeah. So that's usually a way, because you can't do in a lab. You're not going to do clinical studies right away. So you'll start with
I understand that, because you
human
can
cell
do
line.
without an efficacy study. You can move
Yeah.
forward, which is
And
totally
plus, if
understandable.
you're testing a new ingredient, you want to first test for what dose level you are considering. So you do a dose response curve and things like that in a human cell line. And then based on that, you can start figuring out, okay, these are the concentrations we're thinking of using in our formulations. And things like that.
No, that would be a good case study, but then given this is going to be also a topical ingredient, so for me, I feel it's going to be nice if we can look up into the serum samples of the patients, which would give you probably a better benefit moving forward. So
Yeah,
was
and that's what
follow-up
they do
question.
when they move into the clinical study, is they start looking at the clinical endpoints. So things like we have standardized tools for clinical studies, like a corneometer and things like that will test hydration levels, tool levels, things like that. So as we move from in vitro, where we look at some of the benefits, then we go into the clinical endpoints, where for redness, it might be a dermatologist doing a clinical grading and things like that. And then for hydration, you have these handheld tools like those.
Thank you, Kavita, for great questions. Those were amazing questions, and they gave a lot of information on how the research works. Akshat, we would highly appreciate if you can put remaining 2 links in the chat. And if anyone has any other questions, please feel free to go ahead and ask the questions.
I think I'll drop now because I have to join my other call. Okay. Thank you, Akshat. Thank you, Kriti. And thank you, Kavita. And thank you, Nidhi.
Thank you so much. Thank you.
you.
Great talk, Akshat.
Thank
and
you.
thanks, Pratibha.
Thank you.
Thanks. So thank you everyone for joining us for this presentation, and we would highly appreciate if you can consider volunteering for GCAR. As I mentioned earlier, that any nonprofit will only be successful with the help of the volunteers. So we would highly appreciate if you can consider volunteering for GCAR. And with that, have a great rest of your day.
Thanks.
Thank you.